Buongiorno a tutti.
Ladies and gentlemen, good morning and welcome to this meeting that is devoted to one of the most important topics ever, so brain and consciousness. It's beautiful to have an applause for the brain. So, it was really impossible not to tackle this issue during this 40th edition that has this title. So, your name was born from what you gazed upon, and this line taken from a poem written by John Paul II expresses the essence of human beings. They stem from relations and then they develop through relations. So relations are key for humans, and while cloning is certainly not a good way for human beings. And so it goes without saying that science is trying to better understand a specific aspect of our humanity that is certainly the most fascinating aspect of our sort of essence of our entity, that is intelligence, so the brain. Yesterday we had a wonderful meeting about artificial intelligence, and Professor Magazzini, an expert in artificial intelligence, said that the biggest wonder ever is the human brain, and that struck me a lot. So he didn't mention, artificial intelligence. He didn't mention the world Go champion that was beaten. The sort of less important element. So it's not the computer that was most important, the one that beat him, not at all, the other way around. And then I was struck also by transhumanism, transhumanism, and Mark O'Connor wrote a book about it. And what strikes me the most is that this movement is not based on scientific facts, on evidence, but simply on a sort of principle, considering the human brain as a computer, as a personal computer, but actually the whole science seems to prove the opposite. But, well, if you build a theory on the basis of prejudice, well, then sooner or later the reality will debunk that theory. Today we have great speakers, two international scientists. We're very humble, and little by little, very humbly, are trying to investigate the functioning of our brain. How does that work? What are the processes? How is our personality sort of likely to express itself through our brain? Because everything goes through the brain. Every moment of me goes through my brain. I can't be sort of detached from my brain. My self, my subjectivity is something that is deep, deep down. It's a big mystery, like a black box. And I think that our speakers agree with this idea. So, but again, it's about trying to understand the wonder of these beautiful thing that is our brain. And the more we're going to investigate, the more we're going to discover about the beauty of the human brain and of human beings. We have Professor Egidio D'Angelo today with us, who is full professor of physiology at the Department of Brain and Behavioral Sciences of the University of Pavia, and is the author of Cellular Physiology, a handbook for university students, and is also a member of the Scientific Committee of Human Brain Project, that is a European project that has been financed with 1,200,000,000, that has been going on for quite some time. It's a sort of a 6-year project, and the aim is to sort of create models of the human brain, so to try to understand which are the processes and create model on the basis of those processes to better understand how the brain works. So it's going to tell us more about his work and his future plans. The second speaker of today is Professor Vittorio Gallese, full professor of Psychobiology at the Medicine and Surgery Department at the Neuroscience Department of the University of Parma, and he comes from the Rizzolatti School, so that's his background, and this is something great Italy can be proud of, because this is the school that discovered the mirror neurons. Well, I guess that you know them. And when we see another person who is doing a certain action, well, specific neurons activate inside ourselves, and that are close to the neurons that we would use to make the same gesture, but they express also some form of empathy. And I'm going to ask him some questions. And I forgot to introduce myself. I am Mauro Ceroni, and I'm a neurologist, and I'm also a professor at the University of Pavia. Wow, it's wonderful to have so many applauses. And I work at the Mondino Institute, and I'm one of the coordinator also of the Brain Space. You're all very welcome. Please do come and visit us. So there's plenty of nice surprises. But now I would like to give the floor to Professor Egidio, who is going to tell us more about his work.
Buongiorno.
So good morning everyone. Thank you, Mauro, for his kind invitation. This is really a great honor and a great occasion to be able to participate in this great event. My task today is to give you the point of view of a neuroscientist, and in my capacity as such, to tell you what we are learning and what we have understood about the brain. In the introduction, Mr. Ceroni talked about what was said yesterday about artificial intelligence. I think this can be greatly enriched and enhanced by future knowledge about what is taking place in the brain. In other words, understanding the functions of neurons and the mechanisms with which the circuits of the brain work. The brain is an organ of the human body, and it is not by chance that I decided to use this wonderful picture taken by the Code by Leonardo da Vinci, in which he studied man and the brain. So the study of the brain has in its very first evidence of study these works by Leonardo. As a matter of fact, Leonardo, starting from his studies on the brain, was capable of understanding the inner structure of this organ. Many unknowns still remain about the brain. First and foremost, when the brain circuits enter into force, what they calculate, because we know that these circuits produce calculations, and how possible it is that they make these calculations. In other words, the when, the what, and the how of the brain function. Today's neurosciences have greatly changed compared to the past. At the moment, their knowledge stems not only from the clinical observation of patients who suffered injuries from the brain, or anatomofunctional or pathological characteristics of the brain. Neurosciences today use tools coming from mathematics, from biology, physics, or chemistry, as well as other subfields. So the first thing to consider, and I would like to be as intelligible as possible in telling you this, is the following. Like all bodies, organs of the body, the brain is organized in a sort of hierarchy of complexity from the neuron to microcircuits made up of a number of neurons which are about tens of millions. And from that we go up to microcircuits which are made up of these macro assemblies. In other words, sets of macro circuits all related one to the others. And this is the level of the entire brain. And the extraordinary thing is that the brain does not bring about a mechanical action like the heart, for example, or the muscle, but rather a behavioral kind of behavior. So it makes it possible for us to think, to act. So what we measure as an output in terms of brain function cannot be measured with a normal device that might be used to measure, for example, a dislocation or a force or a movement. This brings about a series of circumstances that make it difficult to study the brain, and hence two fundamental approaches derive. One is the bottom-up approach, in other words, from the bottom to the top, to the top. So one starts from very detailed analysis of the molecules, of the subcellular structures like the ones you see here, so the synapses or the neurons. This approach is very powerful. However, it has one limitation. In other words, up to over a certain level of complexity, actually you no longer understand. So you stop understanding. And then you have the opposite approach, a top-down approach. You may start from a clinical analysis, from a psychometric analysis. You may start from the analysis of nuclear magnetic resonance signals. And from that, you try and understand what's taking place in the brain. Of course, you learn a lot of things, but you're very far away. You're very distant from the neurons. So you do not understand what the neurons underneath do. Both approaches are very interesting. They've been widely deployed by billions of scientists, but they do not correlate, they do not communicate, and this is a critical issue. So what do neurons do? Neurons are cells. They have all the properties of all the cells in the body. They have a very special property which is amplified. In other words, they generate electric signals in the cell membrane. These electric signals are action potentials. They are called action potentials, and you see some representations of them. So action potentials are depicted as some kind of spikes, and spikes get organized over time in sequences, and sequences contain a code. The same code contains the information. We've often heard about codes and information also yesterday. These are the codes of the information of the neurons, in other words, sequences of action potentials. You see here two different neurons, the granule cell and the Gallese cell. But what's interesting here to note is that the spikes get organized in regular discharges or in bursting. They can generate slow oscillations like the ones that you see here. They can generate a self-rhythm. In other words, these neurons are pacemakers. They work by themselves without being stimulated. They can even generate and stimulate reversed phases of polarization, the so-called hyperpolarization. They can produce a frequency, the so-called adaptation phenomena, or they can generate pauses and some rebounds. In other words, they can have discharge rebounds at the end of the pause. All these behaviors are called non-linear dynamic behaviors. What's interesting to note is that the brain neurons, and there's some hundreds of phenotypes, are not always the same. From this point of view, they behave differently. When we move from the investigation of neurons to the investigation of their sets, in other words, of their circuits within which they are connected through synapses, some interesting things emerge. Within a microcircuit, like the one that you see here on the right, in the bottom right of the picture, neurons do not do the same thing. They have a diversified discharge and they communicate one with the other. At a higher level, Let's assume that we are analyzing an animal and we are measuring through the field electrode the set of the neurons of the animals. That basically represents the total of all the discharges of all the neurons of the animal. And that field potential is the basis of the electroencephalogram, the EEG. And the EEG is made up of a series of extended field potentials which are extended and measured in different parts of the field. If you go even higher, you can move on to different areas of activity of the brain, and here we use nuclear magnetic resonance imaging. So through the use of MRI, you can see what happens in various areas of the brain. And what you see there is that depicts the flat kind of area of the brain. So at different levels you see different phenomena and you analyze different sets of things. If you go even higher, you may try and understand, you may want to understand what the brain does as a whole. This means the analysis of cognitive functions, emotional functions, movement functions, behavioral functions, attention, motivation, recompensation. Then we have homeostasis, we have the behavior, we have the thought, we have ideation, we have perception. In other words, you should be familiar with all these words. So the physiology of the brain is interested in bringing together what all of these parts and in understanding the interactions between all these functions. A critical issue at the basis of everything lies in the fact that the brain always creates its own internal virtual reality. We live in a virtual world, which is our own world. My own world is different from yours. It's very similar to yours, but it's not the same. So each and any one of us in this precise moment understands something different because we have our own unique internal world made up of his or her own beliefs, internal beliefs. We in a way perceive what I say, then compares what I say with what we have inside, and then compares this with its own beliefs and produces something that is meaningful or not meaningful to him or her. The internal world of the brain is perfectly autonomous, and it compares with the external world through the senses. This is fundamental. And I'll tell you something more about this interaction between the internal world and the external world and about the structures that control this interaction. From the point of view of evolution, it goes without saying that this type of construct brought about very important advantages to all animals that have a brain, not only man. Among these animals, among these beings, man is capable of handling symbols, and symbols can become abstract realities. Man is capable of generating a thought, and that thought can become abstract. In other words, this is a cascade behavior. We have an inside world, and this inside world elaborates a content, and that content is constantly compared to the outside world. When this matching disappears, we have thought anomalies like the ones of psychosis, in which the internal world is no longer fine-tuned with the outside world. From the point of view of system physics, this type of functional organization is called the adaptation or adaptative complex system. Complex because it's based on interactions, and adaptive because it can be modified based on interactions. Let me give you a very simple idea. Just imagine that the brain operates through schemes, internal schemes, and that it uses. So when I utter a word, your scheme expects me to say another word that is in logical relation with the one I've just said. So you expect this word to come out, and that word is not pronounced. If it is not pronounced, there must be something that is wrong. First of all, I raise your attention. There are brain mechanisms that raise the attention based on the unexpected, based on the unforeseen. So the new word that I say is related again to the scheme through our senses. So the scheme starts maybe thinking that maybe the second word I have uttered might be foreseeable. And if this happens again and again and again, that means that after the word A, you always have the word B follows. That is the learning mechanisms. We're learning from the environment. The system is complex, and at the same time it is adaptive. It adapts to the system. All the rest stems from this. And I'll give you additional examples on this. After all these things, so we study neurons, we study the behavior, we study schemes, but we still do not understand how the brain works and what the critical points are. The critical points lie in the fact that the brain is made up of very small elements, that is neurons. The man's brain contains about 1,000 billion neurons. These neurons have complicated, complex properties. They're not equal one to the other. They have action potentials and so on. They interact one with the other, and this brings about complexity. So when we talk about neurons, we cannot understand the whole brain. When we study the functions of the brain, we don't know what the neurons do. And here lies the problem. We have molecular and circuit and structural complexity, and this complexity is enormous. Then we have stochastic mechanisms. Some phenomena are not foreseeable per se, but just for the very same probability that they can occur. In microcircuits, the signals that come from the inside and the outside of the brain get distributed in a very complicated way, and we do not know how this happens. In microcircuits, the information is processed, but we do not know how. At the level of systems, the relations between the structure of the circuits, their functions, their dynamics, and their control is still largely unknown. And plus there's a huge problem, which is the fact that the relation between the brain and the mind and conscience are autonomous. So they are detached from our ability to describe them for a number of reasons, and this brings about additional complexity. So, to sum up, in scientific terms, we lack a model. We don't have a model based on which we can foresee how we can understand the brain. In other words, we should not enter ontological loops here, but what I'm going to say is that we attempt to understand the brain, but we lack a model to do it. And so our task is to build the model. So this brings me back to the Human Brain Project. Some years ago, this project set itself to solve this problem. In other words, its aim was to create a model of the brain to connect the micro level with the macro level. A model can be tested. We make a simulation, and then if we run an experiment and things are different, that means that the model was not complete. So the model should be changed. That model becomes our basis of knowledge. The model can be falsified and it can be proved. And in this way, we can, in a way, build our own understanding of the brain. And then there's additional practical problems. Our knowledge is fragmentary. In other words, we do not know anything. We do not know everything, sorry. So the model is telling us what is missing. If we have lots of details to understand, but if we have time and resources only to understand fewer of them, actually the model suggests us the first 1,000. In other words, because of all these reasons, we decided to work on a big project to study the brain. So how should we proceed? Within the Human Brain Project, we thought about adopting a reverse engineering approach. In other words, we are building the model. Our aim is to try and understand how, for example, a bridge can stand. But if we do not know the structural equations, we need to, in a way, rebuild the bridge piece by piece, reassemble it, and then from the ensemble, from the whole, understand the entire system. And this is exactly what we're doing with the brain. So we are rebuilding the neurons, we're simplifying them, we are inserting them in the microcircuits, and then we are connecting the microcircuits and large-scale models, and from these models we then achieve models of the brain. This is feasible. This is feasible, and we are really doing this. And I'll show you how. There's a coincidence in this respect. This approach can be pursued now. It could not be pursued 15 or 20 years ago, because now the IT is giving us the high-performance computers which make it possible for us to develop such models and simulate them. This was not possible before. Another important fact is that there are huge data banks containing structured and organized information that can be used in the models. This was not available in the past. So once again, this is an IT development. To understand the model, it is not enough to be a biologist or a doctor. That's not enough. We need information, and we need kind of structures that bring together certain and different disciplines. We need interdisciplinarity. So neurosciences are a transdisciplinary approach. So these are models based on differential equations which show in a detailed way what the molecular properties of the membrane. The transitions are represented by differential equations. The differential equations describe how the system behaves over time. And this is therefore the dynamic behavior of the systems. I'll show you an example. What I'm showing you now has already been shown in the area brain. This is the result of one of our works. This is the model of the biggest cell in the brain, the Purkinjean cell. To represent this cell, to do this, we had to use more than 1,500 electrical circuits. Each and any one of them is represented by some dozens of differential equations. So this model contains about 65,000 differential equations. This gives you an idea. This is the detailed model of a neuron. This is actually what a biophysicist or a biologist or a biologist should do to represent a neuron. And this model works exactly like a real neuron. So through reverse engineering, we transferred biology into a physical model. There are spikes, there are bursting, there are pauses, all the actions of a neuron. This activity, if you look at the colored part of the picture, this activity is well distributed in all the compartments, and the yellow parts are the action potentials. Now I'd like to exemplify a little bit, and I'll show you some examples. These examples are applied to the cerebellum, which is that part of the brain that is in the back, which for many years has been considered to be actually the repository of the controlled movement for men and animals. Actually, listening to the musician would be more interesting than listening to me. You've probably seen how her body behaves, how her hands behave, and how the musician is actually leaving her world of sounds and transforming this into an action. So what the cerebellum does is to coordinate the several movements of her own body. She kind of coordinates them. The cerebellum coordinates them. It generates the spatial and temporal relations of 600 muscles. So from a mathematical point of view, we are working in a geometrical hyperspace, which is organized around hundreds and hundreds of axes. Instead of having only two axes, X and Y, you have, let's say, 10,000. So what this organ, this internal organ of the brain is doing is extraordinary. And the question is how? How can it do it? In future studies also showed that the cerebellum does the same action not only in coordinating movement, but also in coordinating thoughts. Our thought is coordinated logically as if it were a movement. So when animals learn how to move, they learn to understand what they do. They first think, they reason, and then they move. So movement is the enactment of thought on behavior, on actual behavior and movement, but actually thought and movement are based on the same circuit and neurons structure. During evolution, the brain has been increasingly connected to the parts that control thought. Cerebellum is organized in modules. There are lots of modules. They are more or less, they look the same, but they connect to different parts of the brain cortex. So here you see an example of the reconstruction of the connections that was done with MRI, so nuclear structural MRI, and then tractography. And this is going to be illustrated more into detail by Claudia. This study analyzes in detail the connections of the cerebellum modules with the cortex. This is an example that shows how the cerebellum functionally connects with parts of the cortex and pulsates with them. So this study is done with magnetoencephalography because it is not possible to carry out an EEG of the cerebellum. So the another interesting thing is that the cerebellum participates in cognitive control phenomena, and in this way, it actually controls other parts of the what people are doing. So basically, the cerebellum is getting these pictures and simulating them internally. It is sort of teaching the person who's watching what the other person is doing. This is a correlate of the mirroring effect that was mentioned before. Furthermore, the cerebellum participates in the so-called resting state networks that all scholars of functional MRI scholars do. What is out is only the DMN or default mode. These basically regulates our participation in the internal state of the brain, its internal reality, virtual reality, and the connection with this is connected with the attention networks. When an attention signal arrives, we start from our own internal world to the external world, because the external world is giving us signals, and we have to capture these signals to, in a way, understand, defend ourselves, and also interact with the environment. The cerebellum also takes part in the release control of dopamine, which controls our emotional and cognitive states. So dopamine does not only control on the VTA, it is not only connected with the base ganglia, as is normally written in textbooks, but the dopamine system is controlled by the identification of novelties, of errors compared to expectations. And this is something that is controlled by the cerebellum. This has been recently published on Science some months ago. So basically, we're dealing with a structure that shows an increasing integration with the functions of the brain, which are much more manifold. So in the pianist, the cerebellum is not only coordinating the action of the muscles, but also the entire interaction of the pianist, of the musician with the environment, and her entire cognitive and emotional production. So it is a totally integrated structure with the rest of the brain. To study this type of phenomenon, we use models, and we will soon talk about this. What the cerebellum is doing now is perfectly in line with the complex system that I've shown before. The cerebellum learns how to predict the temporal connection between events, be they movement events or non-movement events, so cognitive events. So I'll show you this picture very briefly. The first idea about how this could work was developed by Mar, Albus, and Ito, three scholars who developed the motor learning theory. The first theory that tried to bring everything together. This dated back to 1969. This theory was very interesting, is still considered to be a reference point. However, it was lacking on a number of parts. For example, on where learning is deposited. So the synaptic plasticity, which is at the basis of learning, then the theory lacked information about the discharge mechanisms of the neurons, and plus, for some reason, the geometry of neural connections inside the circuits were not known. So somehow these scholars understood something, but much information was missing. And without this information, it was not possible to have a model. So their theory remained a theory. This was not actually transformed into a model, and therefore the theory was never simulated. So theory, model, simulation. These scholars only had the theory, they lacked the model, and they lacked the simulation. When their theory was turned into a model, the model was simulated, this is what happens. This is exactly what we're currently working on within the Human Brain Project. Human Brain Project is not something that came into the mind of someone only 8 years ago, but it dates back to 1995. Some projects were under our supervision in Pavia, others were organized and in other parts of Europe. So from the neuron, we started developing neural circuits. These neural circuits were transformed and simplified and inserted within robots or brain simulators. And this again in relation with the experimental data that I've already shown to you. This slide shows that we can transform the very complicated models made up of about 60,000 differential equations into models that contain only 3. Of course, something gets lost in the process. Some of these losses can be made up for, but what's important is that the oscillations, the resonance, the bursts, the rebounds, the adaptation, and the pauses can be kept. So the dynamic behavior of the neurons is still there. We are not losing the dynamics of the neurons. The simplified neurons are then inserted into rebuilt microcircuits because over the years we have developed the IT tools to do this. We have also rebuilt the graphical routines to see them, and this is very important. This is not at all of secondary importance because you have to see what is being done. And we've come up with this kind of representations. This is a microcircuit of the cerebellum that is working. So this microcircuit is made up of some hundreds of neurons, but the real one from which this tiny part was extracted, contains about 400,000. 400,000 more or less corresponds to a functioning module of the cerebellum. In mice, there are about 35 million neurons in the cerebellum, and we have billions in man. So with one of these modules, we can create a very large part of the cerebellum of a mouse and a smaller part of the cerebellum of a man. If you multiply the modules and connect them correctly with the cortex, you can simulate the brain. Let me give you an example of propagation of these modeling effects. Let's take the neuron that I've shown at the beginning, the granule cell. The granule cell is apparently simple. It is apparently one of the simplest to be found in the brain. However, it is very complicated. It is very complex. So to be able to understand how it works, we had to develop a first module that takes all the currents of the brain membrane into account that make it possible for it to work. That's the 10th planet of the sun system. This is because there were 9 and we discovered the 10th. We found it experimentally, and that's the first. Then the model predicted the action potential, but it could not predict its width because the channels were not in the right place. The ionic channels were not in the right place. We then moved them in the right place, and that was the second step. After this step, the propagation was not correctly propagated along the axons, which are the kind of connections from the neurons to the other cells. And so from this, we developed yet another model. So work is constantly being done. And then at the given step, we discovered that the neuron lacked yet another property. So we basically used the model to find the 11th planet. And we discovered yet another ionic channel inside the model. So we are using models to predict biology. We could have looked within the hundreds of ionic channels in the brain, but we could have probably not understood that there was some missing. The model predicted what they had to be. We looked for them and they were there. So models are extremely powerful when it comes to prediction. They restrict the experimentation field to something feasible and also economically accessible. Instead of spending lots of resources to make a lot of experiments, we made a more limited number of resources of experiments, and we immediately found the answer. This example shows the propagation of the effects at the level of circuit. That's yet another representation, and this is a video. And in this representation, once again, we have the activities of the neurons and of the cerebellum within a system of some hundreds of thousands of the neurons within the module. So the video is very nice to see, but interestingly, when you analyze, when you investigate, you discover 2 structures within the data. The first structure that you discover is the one I'm highlighting now, and that's the center surround. The center surround, that's a red central area surrounded by some kind of a blue ring around it. This structure is naturally formed inside the circuit when we stimulate it, because of the properties of the neurons, because of their connections. So this means that channels are being formed, and through these channels the signals are transmitted. The red part is the transmitting part, while the blue part isolates the signal from the basic background noise, basically. Here we have another structure, and that's the correlation between the activity of the neurons. When we stimulate the net, the neurons present a correlation one with the other. So they pulsate synchronically along predetermined wavelengths, and they are dictated by the oscillation capacities of the neurons and their connectivity. So this system of models is predicting what the circuit is doing. Then we make the tests, and we can find both the oscillations and the center surround that you see here. So we've been capable of predicting what the circuit had done, and we've seen what it really does, and it really does it. This last example shows what happens when we enter learning, so the synaptic plasticity. So we basically transfer the circuit inside the brain. I mean, learning per se and by itself is useless if not inside a brain. And so in this case, we are basically introducing the rules of synaptic plasticity, and we are having these experiments inside the neural robot. These experiments were carried out in the lab run by Alessandra, and she's going to talk about this aspect more in detail. The neural robot contains the circuit of the cerebellum. Its neurons inside the circuit are working exactly like in the simulations that you've seen before. And the interesting thing is that the circuit is learning. It's learning through the rules of synaptic plasticity autonomously. In other words, we did not tell him what it should learn, but rather only what it should do. And then the rules of the action it should do are acquired from the interaction with the outside. From an IT point of view, information or energy is being transferred from the outside to the internal circuit. It is exactly doing what the brain is doing. Artificial intelligence is not something to be mentioned here. The artificial intelligence is just a construct of the engineer who writes rules and implements them. Here we're not writing any rule. We're only telling the circuit what it should do, and he does it by itself. And so there's a big difference. It's not artificial intelligence. This is not artificial intelligence. So you can see here a long time how learning takes place over time. So as long as the exercise goes on, this discharge changes. You see this blue stripe. This means that the neurons are silent. They are no longer discharging. And then you have this very deep cerebellum nucleus, which received the signals from the stripe above, and they were silent before and they stopped functioning again. This is a key paradigm of the way in which the cerebellum work, and this was based on the theory mentioned before of the three scholars. So the robotic models are confirming their theory. So the system is learning to correlating events on a millisecond scale. This is a very nice example developed in one of Alessandra's labs. What you see here is the circuit of the cerebellum introduced inside of a humanoid robot. This circuit helps now, that's the name of the robot, to find a correlation between a harmful stimulus, which is this laser beam directed towards the belly of the robot, and a sound. So the robot is learning to correlate the laser beam and the sound, and just see what he learns to do. So basically, he generated an internal correlation which is basically written as synaptic plasticity in the circuits of the model I've shown before. And he's writing the temporal relation between the sound and the laser beam. And then once the sound is identified, there is immediately the corresponding adequate movement. Of course, we had to tell him what reaction. But what's important is that the cerebellum is learning this temporal correlation. It is as if our pianist actually has learned that after a given sound, he has to move the other finger to generate yet another sound. And then sound after sound, you generate the entire scheme of the beautiful Chopin piece that you heard before. There's one problem that still has to be solved. Actually, up to this moment, we've always gone using a bottom-up approach. But what happens if we wanted to understand what happens if we were to understand the signals generated by an MRI or an EEG? There's a huge problem here. With a bottom-up approach, we arrive more or less at this level. But we cannot reach the very top. That's too complicated. But in fact, the signals coming from MRI reach us independently. Actually, we don't want to build them. We want to see what's taking place in a real brain, in a real man. But we have to read them and understand what happens to the neurons generating these signals. From a mathematical point of view, this is an inverted problem. So we have an ensemble signal that was generated by some lower components, but we don't know how. And we can make an example, a comparison with the physics of stars. Just assume that we've made that you have a map here. This is no longer the activity of the brain, but rather the heat on the surface of a star. You probably know that this derives from the nuclear reactions inside the star. That can be described. So in physics, very precise relations were created between the behavior of atoms and the consequences, the macroscopic consequences on the matter, so on the matter of the star. So in physics, you can understand this map based on the behavior of the atoms that are below. But from the point of view of the brain physiology, this is something that we still do not know how to do, but we would like to. We would like to understand how the neurons below behave in order to generate that energy that is at the top. To do this, one of the approaches that we are using is that so-called Bayesian Inference, which basically solves the inverse problem of going from the global to the activity of the neurons. But of course, in between, you need a model. And what model can we use? We might use a model like the ones that we have seen before. So you see the model, and basically this means really understanding how the brain works. There are technical problems here, IT problems at various levels, based on which we should go from an intermediate representation. In other words, we need to try and transform the neural activity of the model that you've seen before into something that is easier to handle, which is mathematical and written in continuous equations in time and space. I'm not showing them, but we do have them. Basically, what happens is the following. When we do this, we can generate an abstract model, no longer based with neurons that create the field, the action potential, which actually connects the signals coming from the left and actually, in this case, they're basically generating the basic process for the brain simulation. The process has started. We have additional 3 years to complete it, and we hope we will be able to complete it at the end of the project, of the human brain project. It's important to note that all the properties that we have discovered in neurons are in this representation. So the MAR theory, represented by the adaptive filter, is there. We have the center surround. We have transferred the idea of the oscillators, then also of the perceptrons, so the big cells that I showed before that was pulsating, that we also transferred the plasticity rules, and we transferred the connectomy. So basically the science stems from the reconstruction of MRI of the connection traits between the brain and the cerebellum. So all of this is now part of the model of the brain. And this is what we have termed the data-driven model. We are not inventing anything. There are some assumptions, interpolations. We can modify them, but there's nothing invented. So we are working on a data-driven model. At this stage, we can hope we can use this project to really understand how the brain works and develop adequate theories for the functioning of the brain that still do not work. So from a practical point of view, first and foremost, we have an objective. First of all, we have to understand what is happening. But this gives you an idea of how much Europe, the whole of Europe, is spending for the treatment of neurological and nervous problems. in Europe, about 500 billion euros every year are spent. In Lombardy, about 10 million euros. So in line with the costs in Europe. And I'm referring to the social and health expenses, hospitals and treatment. How much does Europe spend for research on the brain? About 5 billion a year, so 100 times less. This data is very important, but if you should divide them by all countries, and this is really little. We all lack. We all complain about the lack of funds. And look at Italy. The declared expenditure and the expenditure which is statistically detectable for Italy is this. It amounts to about 50 and 100 million euros, and that's very little. There are lots of scientists, me, Vittorio, Mauro, many people who work on this. If you divide this expenditure between actually all of us, there's really nothing. So the United Kingdom is spending more, about 300 million every year. And then look at the industry. The industrial sector invests about twice as much or 3 times as much as governments do. And Italy here ranks 5th or 6th. So this means that the Italian industry is investing less in research, in neuroscientific research than other countries. This is statistics that should induce us to think. Our research system should consider these data very much. So in 10 years, the Human Brain Project is spending 1.5 thousand euros. This is a huge amount of money. Still little, however, because actually what we are doing is incommensurate compared to the money that we receive to develop the research. So we really hope that all of this has the results that the world is hoping for. So we really hope that models that can help us develop some knowledge of the neuroscientific structures that we have, so that we can promote the diagnosis and the treatment of neuropathologies and develop biorobotic and bio-IT informatic technologies which are high level. So for example, there's a supercomputing system which is currently creating a series of consortia around the human brain, and there will be the use of the biggest supercomputers in Europe, in Lugano, in Bologna, in Spain, and in France. There are 5 supercomputers. They are already networked, and we are already using them in network. Another massive effort is that of the neuromorphous computer, no longer based on the logic of the Turing machine with a centralized CPU, but rather on circuits which stem directly from the ones that I've shown you. We're developing it in Italy as well. So there's a part on this. We're developing this together with Manchester, because in Manchester we have the elaboration center, the processing center of the Spinnaker Center, which is developing the neuromorph computer. We all hope that these investments bring about some benefits for our society. I'd like to remind you a quote by Feynman. Feynman left a series of very interesting thoughts after him. If we like to think that we will eventually learn something about nature, in many fields we will not go anywhere. We have to be careful. We have to bring everything together. We have to put it all back together, because nature does not know this division. This topic is currently being entirely developed in a meeting that we will take place in Erice at the Ettore Majorana School. And I would like to thank all the people who are working on this. I'm not alone, and they're all listed here. Thank you very much for your kind attention.
Non possiamo non essere pieni di
We cannot be nothing but grateful to our guest for this beautiful picture that he painted and for this beautiful overview, and that reminded us of the most wonderful thing ever, our intelligence, so our cognitive ability that goes through the brain and that let us delve into problems and understand and investigate issues through the scientific approach. And now I give the floor to Professor Vittorio Gallese, Vittorio, and so I'm going to have a sort of dialogue with him and I will ask him questions. So I will ask him to guide us in this journey at the discovery of our participation to events. How do we participate in events? So which are the neural implications and the neural basis for that? So I would like to start with the first question. So brain and intersubjectivity. What did neurosciences teach us over the last 30 years? Well, I started dealing with the brain as a big issue 40 years ago, precisely. It was my second year at medicine, and I applied for an internship at the Physiology Institute, and there Rizzolatti was 40 something with plenty of ideas, a lot of enthusiasm and passion, and is still very much passionate, even though he is a bit older, twice as much almost. So I started working on the spot, making electrodes and working on monkeys. And so I started recording neuron activity since then. So that was the first part of my career. First I was a student, then I was a voluntary researcher. And I remember that at that time, well, intersubjectivity was not a research topic at all. Research topics were very different. It was about outer space and to what extent the brain can contribute to the creation of spatial navigation. I mean, I remember that there was also a special syndrome that was studied, the Mindler syndrome, and then at one point we started investigating another topic. How can our brain be able to guide, for instance, our hands and make them grab objects like this cup, for instance? So I remember that at that time very interesting properties were found, especially by studying our motory neurons. We found out that some of these neurons do not only have motory sort of properties that was rather banal, considering that we were studying the motory system. They also had visual abilities, say the neurons that guide my hand and let my hand grab this cup actually also have visual abilities. And I remember that while studying these neurons, we came across accidentally the famous mirror neurons. So mirror neurons are neurons that are activated not just when a monkey grabs an object, but also when are activated when a monkey sees me grab an object or sees another monkey grab an object. So from that point in time on, relation became a top priority issue to be studied in the lab. Well, something I keep telling my students is the following, and I say it specifically when we deal with emotions as an issue. I hope that at the end of my course you will have far less clear ideas compared with the ideas you're having now and the clarity you have in your mind. And as Egidio said, our basic goal is to reduce the level of complexity and the big mystery and try to reduce it to a number of elements that we can, we hope to be able to do so, to measure. So, in other words, in the lab, we try to reduce the complexity of the world. We're talking about human beings, but actually, we try to reduce human complexity to a set of data that can be measurable and that can be sort of repeated and analyzed. And there's nothing wrong in this approach. I'm a methodological reductionist, and there's nothing wrong with that. But I get a bit angry when, ontological reductionism tries to replace, this kind of approach I have. So when people say, I am my brain, I am my neurons, I am my synapses, well, then I do not agree, because we are something slightly more complex and differentiated than that. And that is simply because we have so many complex neurons in our brain and cerebellum. Neurons do not get angry, do not love, do not hope, do not feel regret, do not promise. No, all these are features that get a meaning when we sort of assign them to the owner of the neuron, so to the individual, to the person. One of our biggest bets is to try to shed some light onto this mystery, trying to understand how brain activity and body activity. I always talk about brain and body. It's very important. Otherwise, we fall in the trap of talking about the brain as a computer or a sort of data processor, the wonder box, the box of algorithm, and so on and so forth, and all of these sorts of inconsistent ideas. So we are all scientists, neurologists. Fortunately, we have been inoculated with the vaccine, and so we have antibodies against all these sort of kind of delusional sort of set of ideas. The brain is an organ like lungs, like kidney, like heart. What we have not understood yet is to what extent it is wonderfully integrated with the rest of the human organism. So one of our most demanding research topics of the last 5 years is trying to understand the relation between the brain and the heart. So trying to understand that, not just also beyond emotions and their implications. One of the few things I've learned of these last 40 years, I think it's absolutely sort of a fact, is that the brain, the mind of an individual is relational. So, the the relations are key. They're quality and quantity, and relations start long before the birth. Already in the uterus, relations start to be established and developed. If you go back to the gestational period, so for instance, during the 2 pregnancies of my wife, I've done anything on her belly, trying to sort of establish a sound connection with the creature that was developing inside her, with the baby developing inside her. So if you make the fetus listen to, I don't know, a lullaby that was heard during the gestation, or another one that was similar, and you make an encephalogram to the brain of the newborn, you will see that there's going to be a different reaction to the 2 lullabies. In 2011, we published a piece of work where we studied the movements of twin fetuses in the uterus, and we saw to what extent direct movements towards the future little brother or little sister are different from a kinetic point of view with respect to the movements and gestures that are made by the fetus when it explores the internal walls of the uterus or its own sort of body. And then after our birth, we move to another world. so where we can experience also gravity force. And after the birth, parents are desperate, get desperate, because newborn babies like to drop objects, to throw objects constantly, continuously. So we have to recalibrate in this new setting. But even before the birth, our body-brain system make us move differently according to what we're going to touch. So if we're going to touch, for instance, the body of a future little brother or sister, the one we are sharing our first living space with, well, the features of the motion are different. And are the same features that would let us say in a child or an adult that it has the highest level of control. So long before the birth, and it's not by accident that we have encountered this piece of work, wired to be social. So we are wired to be social. So the social side is absolutely crucial and strategic. Everything we learn to be and to become is mostly affected and established by the quality and the quantity of encounters that we can establish, the quantity and quality of the relation that we can develop with other human beings. But how is it that, for instance, we share with a monkey more than 99% of our sort of DNA? Nevertheless, well, well, it's absolutely impossible in the natural world to rank sort of beings and creatures. Nobody can tell who is going to sort of develop in the future. Maybe in the world will remain rats or scorpions or just human beings. But so how is it that by sharing 99% of DNA with monkeys do not set up events like this, do not question themselves, do not make art? Well, as far as we know, a key aspect that, in my opinion, is very interesting, but is not very much sort of discussed, is that we are neotenic creatures. So we sort of get born immature. Our brain, when we sort of get born, is very, very small. It keeps developing, and so it's fully developed at the end of adolescence. So when my brain is fully developed, so neurons can communicate thanks to the axons that are like sort of copper wires, and when they are covered by the myelinic sort of coating sheath, the myelinization process is completed between 18 and 20 years of age. So what does that mean? That unlike chimpanzee and also macaques and apes in general, our brain keeps on developing through social relations somehow. So we are born immatures, and because of that, during the first months of our lives, we totally, completely, unconditionally depend on somebody else. If there is no other taking good care of us, tender loving care, well, we are doomed to sort of die and disappear. So among all social animals, and there are thousands of solutions to set up some form of society, from ants to insects in general. Well, our sociability, so to speak, is pretty unique and different, just because we depend and rely so much on the other, especially at the beginning of our life. One of the few things I've learned over these last 40 years is that our mind cannot be understood if we do not consider the other. And this is exactly the opposite of what cognitivism has tried to teach us over the last 50, 70 years. So everything occurs everything, according to them, seems to be concentrated in the mind of the subject. And so everything relating to relations, according to this false and wrong theory, well, would be nothing but simply, the application implementation in the social setting, the features and properties of a mind that is the mind of the individual. This is not like that, because if we are not able to sort of establish relations with the others, then things get very tough for us. So we are not the same at all. So we can then develop relational problems and issues that can sort of take so many different forms. So let's say that in order to understand the mind of an individual, we need to consider also the relational aspect with the others. And one of the biggest sort of defects related to technological limitations is that the vast majority of neurosciences simply concentrate on individuals' minds. Today, thanks to hyperscanning, we start measuring the level of activity of brains of two people while they are having some sort of relation. So, for instance, what we're trying to do is to see what changes if I go to the movies by myself, all alone, or if I go to the movies with somebody else. So again, these experience sharing aspect is extremely important. This is another key word. So trying to see the difference of our reactions when we experience things all alone and we experience things with other people. And I think that this topic is getting to become more and more important in the future. Is thank you very much indeed for shedding some light on this so interesting topic, and for telling us that the brain of an individual cannot exist all alone, but exists within relations. So relations are today more and more mediated by digital interfaces. To what extent do neurosciences help us understand these new sorts of peculiarities of our contemporary times? Well, I must confess that not much has been done in that respect. But first of all, a preliminary remark, and this is something that I've also written quite a few times, so this is nothing new to me. Neurosciences are necessary but not sufficient to understand who we are. Because, as I said, they simply take snapshots. They measure one of the aspects that characterize us, and this is certainly a necessary aspect, because without brain, there's no mind, so there's nothing at all. But, dramatically, and oddly enough, one of the most important moments in terms of learning about the relation between the brain and the experience was the post-war period, because for the very first time, because of the tragic high number of casualties and injured people, especially people that got injuries because of sort of guns and weapons, well, our sort of research work in the psychoclinic sector let us understand the functioning of some areas of the brain and some motory functions, cognitive functions, and sort of visual and functions. So we were able to better investigate that relation. But again, I say it again, we cannot reduce our sort of mental life, our spiritual life, our sort of private life to the pure mechanisms existing between neurons, because neurons somehow just measure sort of specific things. They know just sodium, potassium, calcium, chloride. So it's very few things, just the salts and the elements that go through them. So this is something very specific. Through and thanks to the technological revolution today, we are having a more and more different relation to reality. Professor D'Angelo said that somehow the brain sets up a virtual representation of the world. Think about the difference between the camera of your smartphone and imagine to shoot a video what you experience, and from time to time to cover the camera lens with your hands. And so you will understand that from time to time you will have gaps, and those gaps in your video will perfectly match the points in time when you covered the lens of the camera with your hands. But well, actually, this is what happens, every day, because sometimes we close the shutters of our camera, of our lens, because we close our eyes and we have not understood yet why do we have to close the eyes so frequently. We need to keep the cornea surface wet enough, but sometimes the number of times we close our eyes depends on the topic we're talking about, the person we're with, the emotions we are sort of having inside us, and so on and so forth. But we are not aware of this shattering of our lens, and then there are the rapid eye movements that make us move eyes continuously hundreds of times a minute. So every time we do that, every time we close our eyes, we get black holes in our ongoing live video of our own life. But why do not we perceive these gaps? Just because our brain and our body make up something unique, a whole sort of entity. But today, for the very first time ever, our relation to the world is strongly and heavily affected by the media. And over the last few years, for instance, there is a very banal example. Well, it's a summertime, it's very hot, so we sweat. Well, what's the temperature today? 38 Celsius degrees. But this kind of things are not enough. We have the temperature, but also we have the perceived temperature. And usually the perceived temperature is higher than the real temperature. So, and usually the perceived temperature is higher, and this is what makes us sweat like hell. So again, why is this digital revolution changing things so much? Well, actually, the answer to this question would be very long. But when it comes to our human relationships and physical relations, let's say, is that we have empathy. Empathy means feeling with the other. It has nothing to do with being sympathetic, being good, being charitable, and I do not agree with what has been said about mirror neurons. So nature makes us good because we have mirror neurons. It's a culture that makes us, mean and bad. So absolutely false. Feeling empathy means feeling with the other, and then being sympathetic means sort of feeling for the other. So this is something completely different, and these are two different things. I can sort of also enjoy some, in an extreme scenario, sort of hurting somebody else, and I could sort of get more joy by sort of being very nasty to another person. And these are reactions that we can also measure because thanks to the discovery of mirror neurons, we found out over the years that emotions like fear, rage, or disgust, pain are mapped into the other through our simulation in our body brain. So, and this is the essence of our basic mechanism. And empathy is not a cognitive mechanism, is not a language related mechanism, it's the general perception of what the other is feeling. I can understand what the other is feeling in many different ways. What is missing in the other ways is the best author on, of the book was written in 1812, and this author, Epstein, writes a PhD thesis on empathy, and Husserl was her teacher. And there's a key extract when she says and asks herself, is it possible to feel empathy with animals? And she said, well, when I faced with an animal that is feeling pain or makes maybe sort of some motion. So Edith Stein writes that, well, she can try to get closer to that animal, but what is missing is what we usually feel and how we behave when we feel empathy for another human being. So the phenomenon is lacking. When I see a smiling emoji, I can understand that the other person is happy, but a completely different thing is to try to do it by reproducing the joy of this person by activating those mirror neurons, those mirroring mechanisms. But there are so many ways to learn, and this is one of the traps of language. So, considering that we have just one word to express the notion of understanding, we think that there's just one way to understand, and this is what classical cognitivism has been telling us for so many years. So, the other seems to be a mystery to be solved, and to access the mystery that is the other, I can simply and just create a theory about the mind of the other. But this is not true, this is false. There are other direct methods, and the discovery of mirror neurons has been key for many, many reasons. And for instance, among the others, it sort of relaunched the topic on empathy, because before 1992, if you have a look at how many people talked about empathy before that watershed date, the discovery of the mirror neurons, you will see that there's a huge difference. Very few before and so many after that date. So we have just got back to an old idea. And I also want to say that we are not very original. I mean, it's simply important to be humble enough to go back to already identified key areas of research and go back to the kind of research, because of course today we all feel very proud of our technology and what we have found on our brain. But let's go back to Giambattista Vico, and many of the things we're saying today, well, it was already saying them in the 1800s, or well, in 1700s, sorry, or without forgetting our Aristotle and the pre-Socratic philosophers. Say, to me, aesthetics is very important today, because we are living in increasingly aestheticized world. So the key role played by neurosciences today is not simply or just about, as I've done so far, to try to understand why a certain kind of movie sort of carries me away, and maybe why I love to read about the Karamazov father and the holster of the family, why I'm just gobsmacked by a painting with a cut by Lucio Fontana, or by the Scrivegni Chapel. This is unique feature of our species, but aesthetics is one of the key sorts of elements to understand our contemporary times, because aesthetics seems to affect anything, not just the marketing of products, but also political messages, but also education. Well, so we have infotainment today, we have edutainment today. So education, information, well, they all have to go through richer and richer experiences, likely to be enjoyable, to be appealing, and lead to a massive liberation of that neurotransmitter that is dopamine. Dopamine, and it was also mentioned by Egidio. Well, just think about it for a second. Even politics today is more and more made of communication, and this communication is coldly and scientifically sort of studied and measured. There is an aesthetic framing of metaphors, words, images. So if we really wanted to get to the heart of our contemporary times, we should concentrate on this aesthetic dimension of social life. And secondly, we have to consider the intermediation element posed by technology. I have a 15-year-old daughter, so I live what I'm talking about every day. Tragically, well, not that tragically, because fortunately she's a big reader and she likes paper books. But I've seen groups of teenagers being in the same place, in the same room, and communicate to each other not by talking, but by using WhatsApp or other social media. Well, WhatsApp is considered as something for older people, and Facebook is considered something for very old people. So this kind of mediation introduces relational modes that were totally unknown until a few years ago. But most of all, these things introduce something that is very interesting also from a scientific point of view, because for the very first time we can hold images in our hands. So there is no remote control. Our remote control is our body, because pictures can be seen for the very first time, well, in our peripersonal space, and so not at a distance of 2 or 3 meters, for instance, when we watch TV lying on a couch, or when we're sitting in a movie theater watching a movie. And the vast majority of video and audio information that we get are enjoyed and experienced by us inside a very special space. That is the proxemic space. Is the space that we defend and protect the most. It's our space. It's the space we usually keep when we talk to another person. And usually there are also differences in the kind of sort of tolerated personal space amount. I mean, usually it's a motory space. This is the space defined by our motory potential. And usually people from the north have a sort of need a bigger space, and we're just unlike sort of people from the south. And so there are many repercussions that come from that. So there are 3 main axes of research, and I hope to activate a 4th one, but the main issue is the lack of funding. So my 3 main research axes are the following. So the role of emotions in intersubjectivity, so post-traumatic troubles after stress. We have been to study the sort of post-traumatic traumas in a piece of research work in Sierra Leone. Okay, I have to draw to the conclusion, and that's why you asked me 2 questions instead of 3, as we had agreed. So we have studied also the relation between trauma and schizophrenia, and I hope to relaunch a new axis of research on language very soon, and then another axis of research is about aesthetics. And many colleagues say, aesthetics? Aesthetics is something you should deal with once you will get retired. But this is such a big mistake, because it's not simply about sort of understanding my reaction when I see a zoomed image or an image shot with a Steadicam, and this has to do with the construction and decoding of communication tools. We need to see and understand aesthetics not just as a limited area that is limited to art. Not at all. We need to consider aesthetics in its etymology, aesthesis. So what does it mean? So we build up a representation of the world that is based on our own sensory and motor relation to the world. And then on top of that, we need to add many other factors. So today, this is absolutely essential. We can't help leaving that behind. So we need to study the aesthetical aspects, not only in terms of context, but also in terms of ideas and in terms of practical testing. And I hope to be able to keep studying these aspects in the future years. Thank you very much indeed.
Mi resta solo il tempo delle cose.
Okay, I just have some minutes to finish with some household announcements. Tomorrow there will be a workshop with our speakers in the brain area. They will be asked questions by experts to delve more into these aspects. This afternoon there will be a meeting with Gandini and Pedrocchi. They will talk about the functional MRI applied to the study of the brain functions, and that will be in the brain area in the D3 room. But there's another household announcement I'd like to say. The meeting is a unique event. For 40 years, it is the result of the contribution by everybody, the speakers, the volunteers, curators of exhibitions, artists, and visitors living it. So everybody gives something of him or herself to the meeting. So the meeting is all of us. And thanks to this, we have exhibitions, we have events devoted to children or to sports, and because of this, they're offered for free to those who live the week of the meeting and those who actually follow it from home. That is why our donations play a fundamental role, irrespective of how much you donate. That's an act of responsibility for the creation of this unique place, so that this unique place can continue existing. So to this end, you will find counters labeled with a red heart all over the trade fair area to donate, and donations will have to be given to the volunteers in these places alone. Thank you very much.